Hello everyone. Today we are going to discuss about the ribosomes. Ribosomes are small, round, dense, granular particle which is composed of ribonuclear proteins. These ribosomes occur freely in the cytoplasm or freely in the matrix of the mitochondria and chloroplast, or these ribosomes may be attached or remain attached to the membranes of different organelles. Like we have seen that the ribosomes are present on the membrane of the endoplasmic reticulum. Due to the presence of ribosomes on the surface of the endoplasmic reticulum, these endoplasmic reticulum are called as rough endoplasmic reticulum, and these ribosomes are also present on the outer membrane of the nuclear envelope. And the main function of the ribosome and the it is till now it has been seen that they are having only one function, that is in the protein synthesis. If we will talk about where occurrence of the ribosomes, we are in both cells, like in prokaryotes as well as in eukaryotes, we are having ribosomes there. So, but the number of the ribosomes vary as per the cell. The cells which are actively dividing cells, which are having actively role in the metabolism, we found their large number of ribosomes. Like we have seen the large number of ribosomes occur in the yeast cells, meristematic plant cells, lymphocytes, embryonic nerve cells, cancerous cells, because they are actively dividing cells. And we have seen that these ribosomes, where there is a more discrete type of cells, are having larger number of rough endoplasmic reticulum. So we can say that their ribosomes will be more in number there. Like we can see, or we can say that it's present in the pancreatic cells, plasma cells, nasal bodies, osteoblasts, chief cells of granular stomach, and thyroid mammary gland cells. So if we will talk about the ribosomal types of ribosomes, we are having, according or as per the size and the segmentation coefficient S, ribosomes are classified into two broadly categories: 70S and the 80S ribosomes. So the 70S is smaller in size, and it's, we can say, roughly estimated that its width is 110 angstrom degree, and the length of the prokaryotic or the smaller ribosome is 290 angstrom degree, while the eukaryotic ribosome is having 200 to 240 angstrom degree width, and the length is the 330 to 340 angstrom degree. Both ribosome, prokaryotic ribosome as well as eukaryotic ribosome, they are composed of two subunits. So prokaryotic subunit, larger subunit is 50S and the smaller subunit is 30S. While the eukaryotic larger subunit, while in case of eukaryotic cell, larger subunit is 60S and the smaller subunit is 40S. So these 70S ribosomes have seen, we are seeing this in blue-green algae, bacteria, and they are also present in the mitochondria and the chloroplast. So their molecular weight is in range of 2.7 into 106 daltons. So if we will enlarge the structure of the small 70S ribosome, we are having 30S, as well as the 50S. So this 50S are having three sites: A site, P site, and E site. These sites, all the three sites, are involved in the protein synthesis. As you can see here, they are involved in the protein synthesis. So now the ATS ribosomes. This ATS ribosome is seen in the eukaryotic cell of plants and animals. They are having molecular weight of 40 into 106 daltons. But their sedimentation value varies in different phyla. So 77S is mitochondria of fungi, 60S in mitochondria of animals, 60S in mitochondria of mammals, 70S in the chloroplast. So if we will talk about the content, how much ribosomes are present in a particular cell. If we'll talk about the E. coli cells are having 10,000 ribosomes, which form the 25% of their total mass. Means जो बैक्टीरिया सेल्स है उसका 25 हिस्सा वजन का कौन बनाता है? राइबोसोम्स. Or if we'll take a contrast to this, we are having mammalian culture cells which contain 10 million ribosomes per cell, which is twice as large as the prokaryotic ribosomes. This राइबोसोम से देखिए नंबर भी कितना ज़्यादा है और उसका साइज़ भी ज़्यादा ही है उसमें. सो नाउ वी विल शिफ्ट टू द स्ट्रक्चर ऑफ राइबोसोम्स. These ribosomes are oblate spherical structure. They are porous, hydrated, and composed of two subunits. The larger subunit is dome-like structure or dome-shaped, and the smaller unit is cap-like structure. So the dome remains always on the lower side, and the cap, the smaller subunit, will come and will make a cap on the larger subunit. The prokaryotic ribosomes are having 70S ribosomes, which are composed of larger subunit, which is 50S, and the smaller subunit, which is 30S. The size of the 50S ribosomal RNA is 160 angstrom degree, and the 30S is 180 angstrom degree. In eukaryotes ribosome or eukaryotic ribosome, that is the ATS, the larger subunit is composed of 60S, and the smaller subunit is the 40S. So we are having two different subunits, larger and the smaller. It has been observed when there is the higher concentration of magnesium ions in the cell, in the cytosol, then these smaller and larger subunits will unite together and form a dimer. If the concentration decreases, get reduced in the matrix or in the cytoplasm, then they remain separated from each other, and that then the structure is called as the monosomes. And when this ribosomes, both smaller and larger subunit, is attached with the mRNA, that time we call them as the polysomes, as we have seen this in the polysome structure in case of in the endoplasmic reticulum, when we was discussing about the protein synthesis. So if we will talk about now what are the chemical composition of ribosome, what is present in the ribosomes, from which they are made. They are made from RNA and the proteins. The 70S ribosomal RNA contain more RNA than the proteins. So we can say prokaryotic ribosomes contain more RNA and less protein. So the RNA content in case of 70S ribosomes is having 60 to 40%, and the protein content is there 36 to 37%. So I have taken one specific example, that is E. coli. E. coli contains 63% of ribosomal RNA and 37% of the proteins. This was about the prokaryotic ribosomes. So now we will talk about the eukaryotic ribosomes. Eukaryotic ribosomes contain less RNA than the proteins. So RNA content in case of ATS is 40 to 44%, and the protein is 56 to 60%. So the eukaryotic ribosome, if we will take the eukaryotic example, the yeast ribosome, which have 40 to 44% RNA and 56 to 60% protein. And the another example of the eukaryotic ribosome is pea seedling, which contain 40% RNA and 60% protein. As we have seen, there is no lipid, so we can say in ribosomes lipids are absent. They don't contain lipid content in them. Now we will talk about the individual subunits, that ribosomal RNAs. The 70S ribosomal RNA is having smaller subunit and the larger subunit. Altogether, there are three types of ribosomal RNAs: 23S ribosomal RNA, 16S ribosomal RNA, 5S ribosomal RNA. The larger subunit is composed of 23S ribosomal RNA plus 5S ribosomal RNA, and the smaller subunit of 70S ribosome is composed of 16S ribosomal RNA. So the 23S ribosomal RNA is having 3300 nucleotides, while the 5S RNA is having 120 nucleotides. And the 30S ribosomal RNA, which has 16S ribosomal RNA, is having 1650 nucleotides. And in eukaryotes, we are having 4 ribosomal RNAs: 23S ribosomal RNA, 18S ribosomal RNA, 5S ribosomal RNA, and the 5.8S ribosomal RNA. The larger subunit of the 80S ribosomal RNA is composed of 3 RNAs: 28S ribosomal RNA, 5S ribosomal RNA, and 5.8S ribosomal RNA. 28, 5, and 5.8 constitute the 60S, that is the larger subunit of the eukaryotic cell. And the 40S RNA, that is the smaller subunit of the 80S ribosome, is having 18S ribosomal RNA, which are having nucleotide 2100 nucleotide. So the 5.8S RNA is intimately associated with the 28S, so these are also referred as 28S associated ribosomal RNA. 28S associated ribosomal RNA. Now we will talk about the ribosomal proteins. So by how we do this thing, how we will estimate how many proteins are there. What we will take the simple ribosome that is composed of 50S and 30S. We will dissociate it by centrifugation. Then allow them to in the gradient, cesium chloride gradient, which is having gradient of 5 molar concentration. So they divide these ribosomal proteins into 2 different layers. One layer is called as the core protein, and the another layer, which are having several other types of protein, they call these as split proteins, simply SP and CP, core proteins and the split proteins. If we will talk about the split proteins, we are having small number of proteins gathered together. They have maybe there will be acidic proteins as well as the basic proteins. It has been estimated that in 70S ribosomes, we are having 55 ribosomal proteins. So the largest subunit, 50S subunit of the 70S ribosome, is having 32 to 34 proteins. As 32 to 34 proteins are present in the larger subunit, so they are designated as L1 to L34. जो 70s सब यूनिट का जो लार्जेस्ट सब यूनिट है दैट इज 50s. दिस 50s जो है इसमें कितने प्रोटीन्स है 32 to 34. इट वेरीज फ्रॉम सम स्पीशीज. सो जो ये इनके प्रोटीन्स है इनको डेजिग्नेट किया गया है L1, L2, L3, L4. If I will ask you L3, you have to simply tell me, ma'am, this is the larger protein of the 70s ribosome. And the 30s ribosome is composed of 21 proteins. So ये smaller subunit है तो हमने क्या इसको designate किया है S1, S2, S3 and so on till S21. So altogether we are having 55 ribosomal proteins. But due to the research, later on they were able to see that the S20 protein is identical to L26. Mean जो हम लोगों ने पहले S20 नंबर लिया था प्रोटीन का that is having identical, that is the same L26. So we had to take one less in S, so we can say that we are having 21 smaller proteins, or S protein is 20. Earlier it was 21, now we can say 30S is made of 20 proteins. And in case of 50 or the larger proteins which include 32 to 34, there is the L protein. L protein they have identified as the L protein. L8 protein is the aggregate. This is formed from the L7, L12 and L10. It means जो L8 है it doesn't exist actually, but they are the aggregates of L8, L12 and N10. So if we will say that now we are having total, we have to reduce it by 2, so we are having total 53 different types of proteins which are seen in the 70s ribosomes. एक हमने कहाँ से कम किया smaller से, because that include in the larger protein. और यहाँ पे हमने देखा जो L8 है it's not a different protein, but it is the aggregate of L7, L12 and L10 protein. So if we have to go for the summary, or before going into the summary, so we are having certain metal ions which play important role in the ribosomes. So we need, we have to, for the proper functioning of the ribosomes, we need magnesium ions, manganese ion and calcium ions. All the three are divalent metallic ions, so they are important in case of ribosomes. So if we will compare the 70s and 80s ribosomes, what we will say? The 80s ribosome is larger than the 70s. So they contain large number of proteins. 70 to 80 different types of proteins are seen in the 80s ribosomes instead of 53 which is present in the 70s ribosomes. The 80s ribosomes have 4 types of RNA molecules, while we are having only 3 types of RNA molecules in the 70s ribosomes. Their proteins and nucleic acid are large sized than the 70s ribosomes. Their RNA protein ratio is 1:1. Several antibiotics such as chloramphenicol inhibit bacteria but not the eukaryotic ribosomes. So there is another antibiotic, cyclohexamide, which are able to inhibit the protein synthesis in the eukaryotic cell. So if we have to go for the summary of all what we said, we can say simply that we are having prokaryotes and eukaryotic cells. So prokaryotic cell is having 70s ribosome and eukaryotic is having 80s ribosome. This 80s is present in animal cell as well as in plant cells. So if we will go into the ribosomal subunit, the 70s ribosome is having smaller subunit, that is 30s, and the larger subunit, which is 50S. The smaller subunit of the prokaryote is composed of ribosomal RNA subunit is 16S, and for 50S ribosomal subunit is 23S and 5S. So if we will talk about the number of the proteins in smaller subunit, it's 21, otherwise its total is 53. We can say this at 20, and we are having larger subunit proteins are 33, so total is a 53. This is the, we have not changed it yet, but actually we have studied it because this is a research work. So in case of the eukaryotes, we are having 80S intact ribosome. If we will divide this smaller subunit and the larger subunit, the smaller subunit 40S in case of animal is having 18S ribosomal subunit, and 60S of animal is having 28S, 5S and 5.8S. So in case of plants, we are having 40S is composed of 18S, and the 60S has ribosomal subunit of 25 to 26S, 5S and 5.8S. So if we will talk about the protein of the 40S, they are having 30 different proteins present in them, and for 60S we are having 50 proteins present in them. So this was all about the ribosomes, and lastly they are involved in the protein synthesis. Either the protein synthesis is occurring in the cytoplasm, or it occurs on the surface of the endoplasmic reticulum. Depends on the destiny and the fate of the ribosomes. If the protein synthesis is occurring, that is the translation is occurring in the cytosol, then protein is destined to the nucleus, or it is destined to the mitochondria or the peroxisomes. Or if the protein is translated into the lumen onto the surface of the endoplasmic reticulum, then this protein is involved in the excretory pathway. So this completes our today's topic, ribosomes. Thank you.